Cells follow different paths of cellular respiration depending on the presence or absence of oxygen. Aerobic respiration takes place in the presence of oxygen. Anaerobic respiration takes place in the absence of oxygen. Cellular respiration consists of 4 stages. Glycolysis occurs in the cell's cytoplasm, and the other stages occur in the mitochondria. Glycolysis, pyruvate oxidation, the citric acid cycle or Krebs cycle, and oxidative phosphorylation. Most cellular respiration in multicellular life is performed by mitochondria, which are found in almost all eukaryotic cells. Mitochondria are about the size of one bacterium. Endosymbiosis theory states that they are derived from a bacterial cell that was ingested and then provided ATP for its host, giving it a survival advantage. The structure of mitochondria is specialized for ATP synthesis. Here is a diagram of the cellular respiration stages. Glycolysis is the first stage of cellular respiration. It involves the breakdown of glucose, a 6-carbon sugar, into 2 molecules of pyruvate, a 3-carbon sugar. Glycolysis means the splitting of glucose. 2 NAD+ and 2 ATP molecules provide the activation energy as they are converted to 2 NADH and 4 ATP molecules. The net result of the reaction is 2 ATP plus 2 NADH plus 2 pyruvates. The first step, glycolysis, is done. It converted 1 glucose molecule into 2 molecules of pyruvate. There was a net gain of 2 ATP molecules. The pyruvate molecules enter the mitochondrion. The citric acid cycle can only process 2 carbon molecules, and pyruvate is a 3-carbon molecule, C3H4O3. Before entering the citric acid cycle, it needs to be converted. Pyruvate oxidation uses the enzyme PDC to convert the 2 pyruvate molecules into 2 acetyl-CoA molecules, which are 2-carbon molecules. Energy is stored by converting 2 NAD+ to 2 NADH molecules. The extra carbons from pyruvate are expelled as CO2. Pyruvate oxidation is also called pyruvate decarboxylation. After glycolysis and PO, 2 molecules of acetyl-CoA move into the citric acid cycle, also known as the Krebs cycle. The citric acid cycle takes place in the mitochondrial matrix, the innermost space within the mitochondria. The diagram shows one cycle, which is due to 1 acetyl-CoA molecule. To account for the 2 acetyl-CoA molecules produced from the 1 glucose molecule, 2 cycles are needed. 1 turn of the cycle due to 1 acetyl-CoA produces 1 ATP, 3 NADH, 1 FADH2, and 2 CO2. 2 turns of the cycle yield 2 ATP, 6 NADH, 2 FADH2, and 4 CO2. Once again, the 2 acetyl-CoA molecules that entered this step produced 2 ATP, 6 NADH, 2 FADH2, and 4 CO2.